Okay, so in this video we're going to look at chapter 15, which is going to focus on innate and adaptive immunity. And if you remember, immunity refers to a resistance to infection. And if we look here, we have innate and adaptive aspects. When you think about your innate immune response, these are the aspects that are always going to be active. They're going to resist infection innately, or sort of independent of antigen recognition. So the innate immune system doesn't discriminate against different types of microbes. Rather, it's going to discriminate against all microbes as a whole and prevent the infection of foreign microorganisms. So some of the aspects of the innate immune response would include things like your epithelial barriers. So the epithelium lines spaces within your body. So when you think about like the lining of your airway or digestive tract or the genitourinary tracts, or even your skin, like those serve as epithelial barriers where these cells line up in a nice row, and because of their intercellular junctions, it's difficult for microorganisms to get into the body. So the epithelial barrier, even though it's just a line of cells, that in itself is part of your innate immune response because it helps to prevent infection. So there are also cells of the innate immune response, things like phagocytes, like the macrophages. These things actually will just target foreign microorganisms, not specific to this type of organism, but rather just foreign. Same with natural killer cells. So these are going to be considered part of your innate immune response. They're pretty abundant throughout your body, and they're just going to remove something that's considered to be foreign. Now we'll talk about the mechanisms of how these cells determine self from non-self, or cells of your body from foreign cells, and that's in future slides here. Now, complement is also part of your innate immune response, and remember the liver makes complement proteins, and these complement proteins can actually form together to form something called a membrane attack complex, or the MAC attack complex. And what this does is essentially makes a barrel-like structure where you have a tube of protein and with an aqueous center or water-filled center, and it effectively punches holes in foreign cell membranes. And what that does is it causes lysis of that microbe or foreign cell. Now, remember, these adaptive immune responses, the innate immune responses are non-specific. When you think about the adaptive immune responses, these are the ones that are specific, right? So this is a different strategy of your immune system, where this is slower to develop, but it more precisely targets a particular foreign microorganism or a particular foreign antigen. And that includes things like your B cells, which can activate into plasma cells. These plasma cells are just large antibody factories, and they produce these little molecular flags of protein called antibodies, which effectively stick onto foreign microorganisms or antigens, and it kind of targets them for removal. Now, it also includes things like cytotoxic T cells. Now, the cytotoxic T cells are another type of lymphocyte, as are the B cells, but these cytotoxic T cells can recognize foreign microorganisms and foreign antigens, and they effectively can either secrete perforins onto that foreign cell, which can punch holes in the foreign cell membrane, or can initiate apoptosis of an infected cell through the activation of caspases, where the cell can actually start to undergo programmed cell death if it's infected with viruses or something. Now remember, the adaptive immune response is learned and specific. The innate immune response is not learned, and it's not specific. So it's actually faster, but very broad as a strategy. Whereas the adaptive immune response is slower to develop, but it's more precise. Think of like the adaptive immune response as being like the sniper of your immune system, right? It's going to very specifically target a particular thing. So in terms of different types of immunity, we're going to talk about innate immunity first. So when you think about innate immunity, remember it's always present, and we attack non-self microbes. But because it's innate, it doesn't distinguish between different microbes. It just basically is just going to target any kind of foreign microorganism. And the aspects of your innate immune response that we've talked about already include the epithelial barriers, phagocytic cells like macrophages, plasma proteins like complement, as well as other cell messenger molecules like interferons. So the adaptive immunity is what targets specific microbes. And the way we know what to target are these antigens. And an antigen is essentially just a molecule that's foreign, right? And these foreign molecules have a particular shape to them, and our immune system can do a really good job of actually recognizing foreign molecules from, non-foreign or self molecules, ones that belong in your body and are made by your body. And so these, cells of the adaptive immune response can target foreign antigens or molecules, and these might be like the surface of a foreign cell. They could be protein that you find inside of a cell. They could be the glycoprotein spikes on a virus. Essentially just really any foreign molecule that can mount an immune response. Now, this type of immunity, right, we talk about adaptive immunity, this only develops after exposure to a specific foreign antigen. And what this means then is that you wouldn't already have an adaptive immune response against, let's say, HIV, unless you've already been exposed to that in the past. And that's the rationale behind vaccination, where if you can give yourself antigens before you're actually exposed to a living organism, then you can prime your adaptive immune response to be ready once an infection occurs, if it occurs. So some of the mechanisms of your adaptive immune response would include things like your humoral immunity, which basically is humor or fluid, and we're talking about antibodies here from B cells, as well as cell-mediated immunity, which are your T cells. We have different types of these B cells and T cells. Now, question here, true or false? Vaccination is an example of adaptive immunity. This is true, right? We talked about how vaccines contain antigens, and those antigens can prime your adaptive immune response to, develop the appropriate cells against those antigens, and then, they can form memory cells, which can activate if you're ever exposed to that antigen again. So the idea here is, well, let's say if you've never been exposed to, let's say, measles before, and if you get the vaccine, that vaccine for measles is going to have antigens that are of the measles variety, right? So they're going to be pieces of the measles microbe that you can inject into yourself, ahead of time, and you can, develop the appropriate cells before you're exposed to measles. So some of the different cells of our immune response include our T helper cells, T suppressor cells, and antigen-presenting cells. We call these regulatory cells because they control the immune response, but they don't actively participate in killing off foreign cells, right? Like these are going to be more indirectly participating in the immune response. That's why they're regulatory. So T helper cells would do things like release inflammatory cytokines, and these things can actually help with the proliferation of new cells, and you really just get other immune cells excited. We also need cells to turn off our immune response. Once it's activated, how do we turn it off? Well, that's going to involve your T suppressor cells or T regulatory cells. And we also have antigen-presenting cells, which are usually macrophages, and these are cells that can, engulf foreign antigens, display pieces of those antigens on their cell membrane, and then present those antigens to other immune cells to kind of get them activated and excited. And then the effector cells here actually are the ones that really carry out the direct attack on an antigen. And so some of the effector cells of your immune system would include your cytotoxic T cells, your B cells, which when they're activated, we call them plasma cells, and they produce antibodies that can specifically attach to a foreign antigen. And we also have different types of leukocytes that can, help to remove foreign antigens directly. So for the antigen-presenting cells, these are going to internalize invading antigen pieces called epitopes. And the epitope is just going to be a piece of that specific antigen molecule. Now, these epitopes are going to be shuttled to the cell membrane surface, and they're going to fuse with this MHC molecule. And what you have then is you're going to have this MHC molecule, which is a major histocompatibility complex. You're going to fuse that with an epitope of a foreign antigen, and then what these cells do is they present that to other immune cells within your body. And these can do things like signal T cells to become activated. Now, a lot of your immune system relies on lymphoid organs and tissues, right? Because the lymphocytes include your natural killer cells, your B cells, and your T cells. Now, technically, your natural killer cells are part of your innate immune response because they're going to non-specifically remove, foreign antigens. Whereas the B cells and the T cells are part of your adaptive immune response because these are going to be cells that develop upon exposure, and they can reside within lymphoid organs and tissues until you're exposed to that antigen again. So some of the major lymphoid organs and tissues of our body include things like your thymus here, which is it's an organ that you find just on top of the heart. It's also in the mediastinal cavity near the heart. and this is most active up until about puberty. Now, the thymus is the site of T-cell proliferation and also T-cell production. We also have bone marrow, right? So bone marrow are where B cells can proliferate and also mature. And this is going to be linked up along a system of vessels, which we call the lymphatic system. So your lymphatic system, one of its functions is to pick up excess fluid from your tissues and effectively carry that back towards your bloodstream, kind of over here by the carotid arteries. Now, this also includes lymph nodes. So if you notice these little darker blebs along the way, those would be the areas where you find lymph nodes in higher concentration. And lymph nodes have germinal centers for B cells and macrophages, and they also have areas for T cells to hang out. So what we're saying then is that if you have an infected region of somewhere in your body, and let's say antigens end up in lymph, well, as that lymph travels through a lymph node, then your immune cells can recognize those foreign antigens and start to mount an immune response. So we're saying that the B cells and the T cells, which are located within a lymph node, as well as macrophages, which are an antigen-presenting cell, these would all start to work together to get the immune system excited to help to fight off that potential infection if one exists. So other types of lymphoid tissues we find are a lot of Peyer's patches. Peyer's patches are large germinal centers of B cells you find in mostly the ileum. And the reason why you have a lot of lymphoid tissue around your small intestine is because you have a lot of bacteria in your gut. In fact, 70% of your immune cells are localized to your gut. And the reason for this is that there's actually more bacterial cells in our guts than cells of our own body. Now, other areas we can find lymph nodes would be the inguinal region or groin, obviously around the digestive organs. They can be also hilar lymph nodes near the hilus of the lung. And these are important because the lung has a lot of air-filled spaces which microbes could penetrate through. So it makes sense to have lymph nodes near your lung tissue. We also find axillary lymph nodes, which would actually receive the antigenic material from your upper appendages or limbs, as well as cervical lymph nodes, which would receive the antigenic material from your head and neck. And all of this converges and brings that fluid back towards your bloodstream, where it drains into the jugular veins, just where the place where the jugular and subclavian veins meet. Now, some of the proteins that are produced by immune cells can be things like cytokines. And so these cytokines can be pleiotropic and redundant. And this means that there's lots of different types of cytokines. There's not just one type. There's actually a broad variety of cytokines. And all of these can have varying effects. Sometimes they can be redundant, which means that different cytokines can have the same effect, or they can actually be pleiotropic, where a cytokine that's produced by a particular gene, if that gene is arranged in a different way after post-translational processing, they can have different effects based on the shape of that cytokine. Now, these are mediators of the immune response. So chemokines are released by different cells, and they can attract leukocytes or white blood cells to a site of inflammation or infection. And we also have colony stimulating factors, or CSFs. So CSFs. And these are actually released into the bloodstream, and they can stimulate the stem cells of your bone marrow to divide and mature, which makes sense because if you're infected with a particular microbe, you'd want to recruit more white blood cell growth from your red bone marrow. So, what we're seeing here is essentially the mechanism by which natural killer cells can recognize self from non-self. And we call this self-tolerance, right? There's a challenge of our immune system to recognize things that are foreign versus cells and antigens that belong in your body. And so the way this works is that, natural killer cells, which are part of your innate immune response, they have activating and inhibiting receptors. And so what these natural killer cells do is they float around in tissues, and they'll, also move through tissues in your bloodstream, and they will bump into the cells of your body, or just cells that they encounter. Now, the inhibitory receptor interacts with this MHC class I peptide, and if this works, then you're effectively not going to kill this cell. We also have activating receptors, right? And so as long as the inhibitory receptor binds with an MHC class I, then these natural killer cells will not become activated, and they will not try to kill off that cell. Now, if this fails and you get a breakdown of self-tolerance, this is what can lead to autoimmune disease, which we'll talk about in a future chapter. Now, let's say if a cell here doesn't have this MHC class 1 receptor, and therefore the inhibitory receptor is not engaged, and the activating receptor is the only one that's engaged, then this natural killer cell will become activated, and it can act to basically kill off that cell. Now, what's interesting in this example is they're showing that not a foreign cell, but rather a virus-infected cell of your body, where the virus actually can lead to inhibition of MHC class 1 expression. And so this is one of the ways that this natural killer cell can identify the cell as either being infected or diseased. And the result of this, that is the natural killer cell, basically can cause the cell to undergo apoptosis, and it will fragment into lots of little pieces. Now, complement proteins are produced by your liver, mostly, and they circulate in the blood in their inactive form. Now, what's really cool about these proteins, remember they're part of your innate immune response, is that they're highly toxic and they can destroy a foreign antigen. Now, the way this works is that they're always circulating through your bloodstream, and so we call this part of your humoral immunity. Now, the classical pathway for complement proteins is that an antibody would bind to a foreign antigen, and it would undergo a process of what we call opsonization, where the antibody kind of targets a foreign antigen for removal from your body. So let's say if an antibody binds to a foreign cell, that can activate complement. Remember, complement can form the MAC attack complex, which punches holes in foreign cells. Now, that's the classical pathway, which requires antibodies. That's a little more complex. It can even be as simple as the fact that there are certain sugars, like glycolipids and glycoproteins, that you find on foreign cells and not your own cells. Remember, part of the plasma membrane are embedded glycoproteins and glycolipids. I kind of think about these as almost being like the gang signs of your body, where like if a foreign cell is flashing up a different glycoprotein or glycolipid or gang sign, what's interesting and cool here is these complement proteins can essentially bind to these foreign glycoproteins and glycolipids, and then form that MAC attack complex that can punch holes in that foreign cell. So if you're flashing up the wrong gang sign, then complement proteins will essentially kill the cell. Now, the alternative pathway here is where essentially these complement proteins can recognize microbial molecules without antibodies. Sometimes there's actually cells of our body that have antibodies just attached, and they use these as receptors. And if you don't have these particular antibodies, essentially the complement proteins can bind and again punch a hole in that foreign cell. So the question here says, where do lymphocytes come from? Well, we know that lymphocytes are the white blood cells. And we know that white blood cells originate from red bone marrow, right? In a fetus, this can come from the liver. The liver actually does have some leukopoiesis ability in fetal development, like before bone marrow exists in a fetus. But in children and adults, really it boils down to bone marrow. So what happens is that these stem cells can divide, they can proliferate and differentiate into different types of leukocytes. And these leukocytes include the granulocytes and agranulocytes. Now, if you remember, the hemocytoblast, which was the major stem cell of your bone marrow, can differentiate into the lymphoid and the myeloid stem cell line. Now, all the lymphocytes come from the lymphoid stem cell line, and that includes your B cells, T cells, and natural killer cells. The B cells and T cells are specifically part of your adaptive immune response. The natural killer cells are technically a lymphocyte, but they're part of the innate immunity. Now, what's cool too is that these B cells, once they're formed and they've divided, they've proliferated, they actually can mature in bone marrow itself. And this process of maturation is essentially where your own immune cells can check to make sure that these B cells are competent, which means that they can actually be activated to carry out a response, and they're self-tolerant, which means they can recognize self from non-self. If there are B cells that are produced that are not competent or are not self-tolerant, then they're destroyed. They're killed off. And if that doesn't work, then that can actually lead to autoimmune disease. Now, T cells, on the other hand, actually mature in the thymus, right? So T cells will move from bone marrow, they'll circulate through the body, they'll travel to the thymus where they will mature there. And again, you're going to have the process of determining whether the cells are competent and whether they're self-tolerant. So from there, once they've actually matured, they can actually move over to lymph nodes or other lymphoid tissues and wait for activation, right? They're just going to hang out in their inactive form. You can call these memory cells. And once they're there, then they can actually, just wait for a foreign antigen to potentially bind to their receptors. And at that point, then they can become activated. Now, just to summarize this process, remember we said that we have the hemocytoblast, which is a pluripotent stem cell here. This can differentiate into myeloid and lymphoid stem cells. This lymphoid stem cell can differentiate into B cells and T cells. Now, if they differentiate into B cells, this is going to occur essentially in bone marrow, right? And these B cells will mature in bone marrow, and then they'll move over to lymphoid tissue from there, like your lymph nodes, spleen, mucosal tissue, like MALT, mucosal-associated lymphoid tissue, which would be like your appendix and your Peyer's patches. But they can also circulate in blood and lymph. And then T cells, these lymphoid stem cells, if they move to the thymus, they can differentiate into T cells, which again are another type of lymphocyte. They're also part of your specific and adaptive immune response. And like the B cells, they can hang out in your lymph nodes, spleen, mucosal tissue, and they can circulate in blood and lymph. Now, what this slide is showing here is essentially how the adaptive immune response can work. And so it all starts here with an antigen-presenting cell. Let's say this is a macrophage. Now, what this antigen-presenting cell can do is engulf foreign antigens, like these could be viral particles, and they can phagocytose those. Now, once phagocytosis has occurred, you can actually chop up little pieces of this virus and into the antigens and their epitopes. And then what this macrophage or antigen-presenting cell can do is fuse that with an MHC receptor and then present those antigens to other lymphocytes, like T helper cells. So what happens then is if this T helper cell has the appropriate receptor for this particular antigen here that the cell is presenting, this T helper cell can become activated. And once activated, it can release something called cytokines. Remember, cytokines were mediators of the immune response. And what these cytokines can do then is go on and activate or stimulate B cells to turn on and turn into plasma cells to make antibodies. Or these T helper cells can release cytokines to activate cytotoxic T cells, which actually participate in directly killing off foreign cells. And what's fascinating then here, if you look at this T helper cell, and it uses these CD4 receptors here, it's a very key aspect of the adaptive immune response. If you remove this T helper cell, then what you've done is you've removed the link between antigen-presenting cells and your B cells and T cells. And this is a very keystone kind of species of immune cells here. Now, the reason why HIV is so insidious and destructive for your immune response is that it infects and destroys CD4 positive cells like your T helper cells. So if you remove these T helper cells, then your B cells and T cells, even if they're there, they can't be excited because you're not stimulating the T helper cells to release cytokines and not really carrying out the immune response in that regard. So remember, B cells can turn into plasma cells to make antibodies, and then cytotoxic T cells are the ones that can actually punch holes or initiate apoptosis in cells of your body. Now, what's interesting here too is that these can form memory cells. So if you look off to the periphery here, T helper cells can actually, some of these cannot become activated, and rather they're going to remain dormant as these memory cells. Some of our cytotoxic T cells will also remain dormant as a memory cell, and these B cells. And the reason why you'd want to have memory cells is that if you were exposed to a virus, and some of these cells participated directly in the immune response, you at least want to make sure that some of these cells can persist into the future. And by persisting into the future, we're saying then that your immune system can store a memory of that infection. That way, if you're ever exposed to this virus again, your immune system response is going to be faster than before. Now, the way that we can recognize antigens by T cells is that our immune cells have receptors that attach to MHC proteins. Now, MHC is major histocompatibility complex. This is dependent on the shuffling of your genes of your body, and because everyone has different forms of genes or alleles, it turns out that we all have different MHC receptors. So, even siblings where you might share very similar genetic material, you're still gonna have different MHC receptors due to shuffling of different forms of genes. And so only those T cells whose antigen receptors fit are going to respond to it. So what we're saying here then is that T cells with a particular receptor type can only respond to that particular foreign antigen. So what we're saying here then is that you will have specific types of lymphocytes like T cells and B cells that have receptors against very specific antigens. And that's why it's part of your adaptive and specific immune response. Now, what we find here then is that our T helper cells are CD4 positive, and that's the type of receptor they can use, and our T cytotoxic T cells are CD8 positive. Now, remember, HIV actually infects CD4 positive cells, like your T helper cells. And what we see here then is that our T helper cells can interact with an MHC class II molecule on things like antigen-presenting cells. And so our T helper cells, because they can interact with the MHC class II, this means that they can recognize molecules that have been engulfed by an antigen-presenting cell. So we're saying that this is actually extracellular material, right? Because these antigen-presenting cells will engulf something that's extracellular through phagocytosis and then present that using their MHC class II receptor with the foreign antigen attached and present that to a CD4 positive T helper cell. Now that differs than the cytotoxic T cells because these actually can use their T cell receptors to interact with MHC class I receptors. Now MHC class I receptors are going to pick up antigens from within cells. So you can use MHC class I to basically present antigens of virally infected cells on a cell surface, or you can present cancer antigens on a cell surface. And it's the cytotoxic T cells that can recognize MHC class I receptors. Remember, MHC class II is extracellular antigens, MHC class I is intracellular antigens, and then MHC-II is recognized by CD4 T helper cells, and then MHC-I is recognized by CD8 cytotoxic T cells. So remember, MHC class I you find on all nucleated cells as well as platelets, and these effectively display antigens on infected cells. Now, they're recognized by CD8 positive cytotoxic T cells, and what we're saying then is that these cytotoxic T cells can effectively, recognize whether a cell is infected with a virus or not. And then your MHC class II cells here, you find on dendritic cells, macrophages, and B cells, and they display antigens that are taken up by your antigen-presenting cells, right? So we say that dendritic cells and macrophages, because they're effectively phagocytes, they can engulf extracellular antigens and then display that on their cell surface to B cells and the T helper cells here, and they can signal immune responses due to extracellular antigens. This question here says, which of the following is true about CD4 receptors? Is it A, they're found on T helper cells? Is it B, they attach to MHC-II proteins? Is it C, they stimulate destruction of infected cells? Or is it D, they differentiate in the thymus? Well, the answer here is that they actually stimulate destruction of infected cells, right? That's the incorrect answer of those three choices because we said that our T helper cells were CD4 positive. They do not destroy infected cells directly, which is why this is not the correct answer, right? Remember, it's the cytotoxic T cells that destroy the infected cells directly. So what's interesting too is that your B lymphocytes in some ways actually serve as antigen-presenting cells. And so what these B lymphocytes can do is use their attached antibody here to bind to a foreign antigen, right? This is going to actually bind to that little attached antibody. And then what it can do is essentially present that little piece of the antigen to a T helper cell. And as long as the T helper cell has the corresponding receptor for that antigen, that T helper cell then can release cytokines. And those cytokines, while the B cell is attached to the T helper cell, can actually stimulate that B cell to differentiate and proliferate into more plasma cells. And we talk about these as being clones of the B cell. So you can actually proliferate lots of clones, and then those can turn into plasma cells, which can release antibodies. So what's really interesting about this process, it's almost like fact-checking, right? We're saying that the B cell already has a receptor for a foreign antigen, right? That's the easy part. But what it does though is it also brings that antigen that's bound and then presents that to another T helper cell as almost as if to say like, hey, is this also what you're seeing is foreign? And if the T helper cell confirms that, it releases cytokines. It kind of corroborates the story of the B cell, and it can tell that B cell, yes, that's a foreign antigen, and they can release cytokines to excite that B cell, and that therefore the B cell can proliferate into clones and then differentiate into things like plasma cells or memory B cells. Remember, memory B cells are the inactive version that basically just reside within lymphoid tissue somewhere, and they just store a memory of that infection into the future. Whereas the plasma cells are the ones that are active, and these are the antibody factories that pump out antibodies against that foreign antigen. So the B lymphocytes have antigen receptors, and once they can present that antigen to a T helper cell, the T helper cells can help signal transformation of that B cell into either plasma cells or memory B cells. Remember, this is done through the release of cytokines, and those cytokines can cause that B cell to basically proliferate into more B cells, but that can actually also then differentiate into either plasma cells or memory B cells. Plasma cells are the ones that secrete antibodies, and the memory B cells actually just remain in the body for a long period of time, and they're just there ready in case there's another infection in the future. So there's lots of different types of antibodies here. The first one we'll talk about is IgG. So IgG is a little smaller, and because it's smaller, it actually can circulate pretty well within your body fluids, and it just binds to foreign antigens. Now IgA is also small, but IgA, because of its size, you actually find in a lot of secretions. So this mucus secretions like your mucous membranes, or even like breast secretions contain IgA as well. And what these can do is actually confer a little bit of immunity at the surface, like epithelial barriers of your body. Or if you're talking about conferring immunity to a child, they can receive those antibodies through breast milk, IgA in that breast milk. Now IgM, I think of like IgM monster being the big one. This is a large antibody. In fact, it has 5 different antibodies strung together, and because of the 5 units, they can actually pull these antigens together into clumps, which we call agglutination, and it makes it easier for phagocytes to remove those antigens. Now IgM circulates in body fluids, but because of its size, it typically only stays in the blood. You're not going to usually find IgM in tissues because it's too large to leave the bloodstream. Now IgD is the type of antibody you find on the surface of B cells, and this is the antibody that serves as the B cell antigen receptor that we talked about in the previous slide. So we talked about how your B cells can use this IgD antibody as the receptor for foreign antigen, and then it can present that foreign antigen while it's bound to IgD to a T helper cell, where that T helper cell, if it can recognize that foreign antigen, it'll secrete cytokines and then cause that B cell to essentially proliferate and differentiate. Now IgE is found on mast cells. Remember, mast cells are the ones that you find within connective tissues, and these are the ones that release histamine, and so they're also involved with inflammation and allergy. So when you think about like IgE antibodies, these are going to be associated with inflammation and allergy, as well as autoimmunity. Now, if you look at the structure of an antibody, what you find are these basically protein chains. So over here, it kind of looks like a Y, and you'll notice here we have our heavy chain here. And if you look up top here, what you find is that the antibody can branch into 2 branches over here. Now, you got the variable region of a heavy chain and the constant region of a heavy chain. Now, what's pretty cool too is you have light chains on the side, but these heavy chains and light chains, which are found at the very tip of the antibody here, these are the variable regions, right? So you have variable heavy and variable light chains, and they're variable based on their shape. Now, what's really interesting then is that based on the protein shape here between the light and heavy chains, it can match the shape of a particular foreign antigen. And if that's the case, then this antibody can stick and bind to that foreign antigen. So we're saying that antibodies are specific to foreign antigens because of the fact that they have a specific shape here. Now, the way they become variable and the way they can be different is that these protein segments here are actually created by shuffling of all the genes in your body. So you take all 25,000 genes, shuffle those together, you can create different proteins here, which are therefore variable, and it ends up being like, trillions of different types of proteins you can produce based on the shuffling of genes. And due to random chance, you're eventually going to produce an antibody that has a particular shape against a foreign antigen. If that happens, then you're going to create B-cell clones, and those clones can form a memory where they can basically just produce copies of just the right antibody against the foreign antigen. Now, our question here says, immune cell creates antibodies in response to antigen, right? So what immune cell creates antibodies in response to an antigen? Is it the cytotoxic T cell, helper T cell, cytokine, or the B lymphocyte? Well, we know it's the B lymphocyte, remember? Because B lymphocytes, once they're activated, they can turn into plasma cells, which effectively, can produce lots of antibodies. Now, in the primary immune response, what we find here is that antigens are presented on MHC-II receptors. This can activate T cells, and then T cells can secrete cytokines that allow for B cells to proliferate. And then these activated B cells, called plasma cells, can produce antibodies. And those antibodies can bind to foreign antigens to target them for their removal. Now, some of these activated B cells can form a memory, right? They stay dormant, and they hide within lymphoid tissue until they're ever exposed to another foreign antigen. Now, this takes time, though. It takes time for plasma antibody levels to rise. And if it's your first exposure to a foreign antigen, it might take 2 to 3 weeks before you have detectable amounts of antibody against that antigen. And this is sort of the limiting factor behind your specific or adaptive immune response, is that it's slow to develop, but it's specific once it's developed, right? Now, the problem here, though, is what if you're infected with something that's so virulent that you don't have 2 to 3 weeks to start developing antibodies, right? Well, the good part, though, is that you also have your innate immune response, which is nonspecific. And that's the one that's going to be, the first line of defense there in terms of epithelial barriers and phagocytes and complement. That's going to be the one that's more active before you start developing antibodies. Now, what's cool, too, though, is that, scientists have actually been able to develop vaccines. And vaccines go back hundreds of years. And actually, vaca means cow. And the reason why we call it a vaccine is that vaccines were first recognized by scientists who essentially noticed that milkmaids who were exposed to cowpox were less likely to get smallpox. And so that's what they called a vaccination, because essentially the milkmaids were exposing themselves to a similar virus that doesn't really infect humans, but it was similar enough to where they can sort of introduce those antigens to their body ahead of time. So once those milkmaids were exposed to smallpox, they already had developed some adaptive immunity against those antigens. And that's why we call it vaccination. Vaca means cow. So vaccinations, though, are a little different nowadays, because what we can do, though, is harvest specific antigens from populations of microbes and then isolate those antigens into a safe solution, and then inject those antigens into a naive immune system of an individual and effectively prime their immune system against those antigens under safe conditions, because we're not talking about living microbes here, or ones that are virulent, rather. That way they can develop memory B cells and T cells before they're ever exposed to a harmful microorganism. And so we call this secondary immune response, right? So we're saying that the secondary immune response would be you've already been exposed to an antigen in the past, so that subsequent exposure to that antigen allows for your B cells and T cells to really mount a rapid response. We're saying that it could be on the order of like hours to days before you start getting a really rapid rise in antibodies produced by the memory B cells against that antigen. So that booster shots cause secondary immune responses, and so antibody levels will be high even before the disease are encountered. So you might get a booster shot, prophylactically if you suspect that you might be more likely exposed to a particular microbe, depending on the condition. Now, the role of helper T cells was, remember, we talked about how they're the ones that are going to be sort of regulators of the immune response, and this is because they release cytokines, and these cytokines can activate other T cells and B cells, but these T helper cells, there's different subtypes. We have type 1, 2, and these type 1 T helper cells can stimulate phagocytes and B cells. These type 2 T helper cells can stimulate B cells, eosinophils, and mucosal epithelial cells, and then this should say type 3, but these type 3 T cells can stimulate inflammation as well. Now, We also want to differentiate between active immunity and passive immunity. When you think about active immunity, this is actually going to be where you're exposed to an antigen and you're actively creating cells against that antigen. Now, this doesn't always occur, though, because you can also have passive immunity. And passive immunity is where you can get some immunity in some form, but you're not training or developing cells against that antigen. So the example here that our book gives is a woman was bitten by a rattlesnake last summer, right? And so if she received anti-serum against the snake venom and she survived, was that anti-serum going to develop active or passive immunity? Well, anti-serum is actually antibodies, but you're receiving antibodies outside of your body, right? Like we're saying that you can develop antibodies in a lab, and you can inject those antibodies into someone's body. That way it can target the antigens of, let's say, rattlesnake venom. That's passive immunity because using somebody else's antibodies isn't going to train your own immune system to fight off that antigen. Now, because she was bitten by that rattlesnake and was exposed to those antigens, that's also going to help develop some active immunity, right? Because those antigens built up in her bloodstream in the rattlesnake venom, and she was going to develop, lymphocytes against those rattlesnake venom antigens. And because she developed cells against those antigens, we consider that active immunity. And then what we say is that this summer she'll be vacationing in the same area. So the question our book gives is, should she get a booster shot against snakebite before her vacation? Like, would it be effective? And remember, the booster shot would only be effective so long as she already had developed some active immunity against that rattlesnake venom. Because if she developed immunity, that means that she has memory B cells and T cells against rattlesnake venom antigens. Getting a booster shot and exposing yourself to those antigens again would actually cause those memory cells to proliferate into mini clones. And because you'd have a lot of these things ready to go, then it's going to help confer even greater immunity. Let's say if she were bit by a rattlesnake again on her vacation, the booster shot would be effective at that point. Now, the booster shot wouldn't be effective if she did not develop active immunity, because what we're saying then is that booster shot would introduce antigens, but if she didn't have memory cells against those antigens already, then you're just talking about the primary immune response. And so it could take weeks to develop the B cells and T cells against those antigens. So another example our book gives here is a woman's baby is HIV positive. Does this mean the baby has HIV? Well, if you're HIV positive, then if the test is saying that you have HIV antigens in your blood, there's a very good chance that you have HIV. The problem though with these tests is that sometimes you can get false positives or false negatives. So you got to kind of take that with some consideration. But depending on the efficacy of the test, it could be pretty accurate. And it's likely that this baby does have HIV if they tested positive. So does it mean the father has HIV or the mother has HIV? Well, I mean, we have yet to determine that. But it's most likely that if this is a newborn, that the newborn probably got HIV from the mother through like placental transfer, or maybe during labor, or through breast milk. So the problem here then is that HIV positive mothers can actually transmit the virus to their fetuses through the placenta, through breast milk, or even during labor where there's like blood and wounds and stuff. Now, the discussion here says, the woman says that since her immunity went to her baby, the baby will not need any vaccinations. Is this correct? Like, would your babies not need to have any vaccines if your mother already had vaccines and had immunity in her life? And the answer to this is no, because you can't give someone you're not going to have placental transfer of active immunity unless you're talking about how maybe like a microbe crossed the placenta. So the baby would need vaccinations in order to develop its own active immunity against different microbes. Let's say like influenza or measles, mumps, rubella, or tetanus or hepatitis. These antigens would need to be exposed to the baby, or newborn rather, in order for that infant's immune system to begin to develop its own active immunity. Now, it's true though that the woman could confer some passive immunity to her infant. And the reason being is that, remember, we have IgAs in breast milk. And IgAs can also cross the placenta. And those antibodies then can confer passive immunity because they can actually start to stick onto foreign antigens, of microbes that maybe make it inside of the baby's gut or microbes that have made it inside of the baby's bloodstream, is that these IgAs can stick onto those microbes. But it's considered passive because getting antibodies from somebody else isn't going to train your own immune system to develop immune cells against that antigen. So another question here says, true or false? Active immunity is achieved much quicker than passive immunity. And our answer here is false, because in active immunity, an individual is exposed to an antigen. That's going to take up to 7 to 10 days before you start developing cells against that particular antigen. However, with passive immunity, it's immediate, because we're saying that antibodies can be created outside of a host, and they're either injected or delivered through secretion like breast milk, or they can cross the placenta, and they immediately confer immunity. So, passive immunity is, good for sort of rapid, immediate types of immunity, but it doesn't train someone's immune system to be able to fight infection or antigen long term. Active immunity is slower to develop, but it's going to persist much longer. So another discussion here says, which would cause the most severe immunodeficiency? A lack of B cells, a lack of cytotoxic T cells, a lack of T helper cells type 1, or lack of T helper cells type 2, or a lack of macrophages. So if you consider the role of all of these cells in your immune response, the question here would be missing which one, or a deficiency of which one, would cause the worst immunodeficiency, right? Well, we talked about how our T helper cells are going to be the ones that are very significant in getting the adaptive immune response excited. Now, macrophages can present antigens to B cells, but B cells are also antigen-presenting cells. So, these can kind of rule each other out in that regard. Now, cytotoxic T cells that, although they're effective at targeting specific antigens, you still have other types of immune cells that can kill off particular microbes. Like macrophages can also remove microbes from a body. They're just non-specific. So really the key ones here then would be the difference between T helper cells 1 and 2. Because what we're talking about then is that if you don't have your T helper cells and they can't release cytokines, then you can't even stimulate your B cells and your T cells to become excited. And this would cause the worst immunodeficiency. And this is the one that's associated with AIDS, right? Acquired immunodeficiency syndrome, because we're saying HIV infects CD4 positive T helper cells. Another discussion here says an individual has type A blood. Which antigens does she have on the surface of her red blood cells? Well, they have A antigens, right? If it's type A blood, she has A antigens. And so another question here says, which antibodies against red blood cell antigens has her body produced? Well, if she has type A blood, then she would recognize any other antigen as being foreign. Like B antigens would be foreign to her immune system. So she can already produce anti-B antibodies against that particular foreign antigen. And so the question here says, what would happen if she's given type B blood? Well, her preformed anti-B antibodies would actually target the B antigens on that foreign blood, and that would cause agglutination, clumping. It would lead to inflammatory immune reactions, which can lead to shock. You can also get blood clots, so it's not good. This would be a mismatch of blood typing, and that could be life-threatening. Now, this clumping of blood, because antibodies can stick onto this B antigen as well, can also plug your kidneys and can lead to renal failure. The next question here says, what would happen if her blood is given to a person with type B blood? Well, that individual's type B blood would have, anti-A antibodies, because that would be a foreign antigen. So the same thing would happen in the other person. And what would happen if she's given type O? Well, O antigen isn't really like an antigen that stimulates an immune response. Really, O is just like the absence of A or B antigen. And so O isn't something that your immune system can attack. It's just a lack of A or B. So an individual with type A blood could definitely receive type O because they're not going to make antibodies against O. And an individual type B blood could also receive type O blood because, again, they're not going to make antibodies against something that isn't antigenic, like it doesn't stimulate an immune response. So think of type O as being naked, right? It's kind of circular here. It doesn't have anything attached to the cell surface. So think about these as being like naked red blood cells. There's nothing to attack, so individuals of a different blood type can all receive type O blood.